Viruses occupy a fascinating niche in the biological world. They exist in a gray area, straddling the line between living and non-living entities. They lack the cellular machinery that defines life as we know it. Yet, viruses possess a remarkable ability to hijack the cellular machinery of living organisms. Viruses are essentially genetic material, either DNA or RNA, encased in a protein coat. This simplicity belies their complex and often devastating effects on living organisms. From the common cold to more serious diseases like AIDS and COVID-19, viruses have plagued humanity throughout history. Understanding how viruses replicate and evade our immune system is crucial for developing effective antiviral therapies and vaccines. By unraveling the mysteries of these enigmatic entities, we can hope to mitigate the threat they pose to human health. This exploration into the world of viruses will delve into their replication strategies and the sophisticated mechanisms they employ to evade our immune system. Viruses, despite their simplicity, have a well-defined life cycle that allows them to replicate and spread within a host. This process can be broadly divided into 6 stages: attachment, entry, uncoding, replication, assembly, and release. Attachment. The first step in viral infection is attachment to the host cell. Viruses have surface proteins that bind to specific receptor molecules on the surface of host cells. This binding is highly specific, much like a key fitting into a lock. Entry. Once attached, the virus needs to enter the host cell. This can occur through various mechanisms, including fusion with the cell membrane or being engulfed by the cell in a process called endocytosis. Uncoding. Inside the host cell, the viral capsid disassembles, releasing the viral genetic material. Replication. The viral genome takes control of the host cell's machinery. It uses the host's resources to replicate its genetic material and synthesize viral proteins. Assembly. New viral particles are assembled from the newly synthesized components. Release. Finally, the newly assembled viral particles are released from the host cell. Herpes viruses enter the host cell through fusion with the cell membrane. Influenza viruses, on the other hand, enter through endocytosis. Our immune system is a complex network of cells and proteins that patrol our bodies, seeking out and destroying foreign invaders, including viruses. However, viruses have evolved sophisticated strategies to evade our immune system. Antigenic variation viruses constantly change their surface proteins, known as antigens. This makes it difficult for our immune system's memory cells to recognize the virus. Inhibition of antigen presentation. Some viruses prevent infected cells from presenting viral antigens. Production of immunosuppressive proteins. Certain viruses produce proteins that suppress the immune response. Latency. Some viruses can enter a dormant state within host cells, making them invisible to the immune system. Antigenic variation is a key strategy employed by viruses to evade our immune system. This can occur through 2 main mechanisms: antigenic drift and antigenic shift. Antigenic drift involves small, gradual changes in the viral genome that result in minor alterations to the virus's surface proteins. Influenza viruses are notorious for their ability to undergo antigenic drift. This is why we need a new flu vaccine every year. Antigenic shift, on the other hand, is a more dramatic change that occurs when 2 different strains of a virus infect the same cell and exchange genetic material. This reassortment of genes can lead to the emergence of a new subtype of the virus with significantly different surface proteins. The 2009 H1N1 pandemic, for example, was caused by a novel influenza virus that emerged through antigenic shift. For our immune system to mount an effective response against a viral infection, it needs to be able to recognize infected cells. This recognition is achieved through a process called antigen presentation. Infected cells display fragments of viral proteins called antigens on their surface using specialized molecules called MHC class I molecules. Some viruses, however, have evolved mechanisms to interfere with antigen presentation, effectively silencing the alarm system that would otherwise alert the immune system. They achieve this by blocking the production or transport of MHC molecules, or by degrading viral proteins that would normally be presented on the cell surface. The human immunodeficiency virus, or HIV, for example, disrupts antigen presentation by interfering with the transport of MHC molecules to the cell surface. Some viruses employ a more direct approach to evading the immune system. They produce proteins that directly suppress the immune response. These immunosuppressive proteins can target various components of the immune system, including immune cells and signaling molecules. For instance, the Epstein-Barr virus, which causes infectious mononucleosis, produces a protein that inhibits the activity of interferon gamma, a signaling molecule that plays a crucial role in antiviral immunity. By suppressing interferon gamma signaling, Epstein-Barr virus weakens the immune response and allows it to establish a persistent infection. Other viruses produce proteins that interfere with the function of immune cells. The measles virus, for example, can suppress the activity of T cells and B cells, two key players in the adaptive immune response. Some viruses have evolved the ability to persist in their hosts for long periods, even for the lifetime of the host, without causing any noticeable symptoms. This stealthy strategy, known as latency, allows the virus to evade the immune system and reactivate at a later time. During latency, the viral genome remains hidden within the host cell, either integrated into the host cell's DNA, or existing as separate circular DNA molecules called episomes. Herpes viruses are masters of latency. Herpes simplex virus, which causes cold sores and genital herpes, establishes latency in nerve cells. The virus can remain dormant for years, only to reactivate and cause lesions when triggered by stress, sunlight, or a weakened immune system. Another example is the varicella-zoster virus, which causes chickenpox. After the initial infection, varicella-zoster virus establishes latency in nerve cells. Later in life, the virus can reactivate, causing shingles, a painful condition characterized by a rash and blisters. Apoptosis, or programmed cell death, is a fundamental cellular process that plays a crucial role in maintaining tissue health and eliminating damaged or infected cells. It is a highly regulated process that involves a cascade of molecular events, leading to the dismantling of the cell from within. Viruses, however, can interfere with apoptosis, either delaying or preventing it to their advantage. By keeping infected cells alive longer, viruses can maximize viral replication and production of progeny. Some viruses encode proteins that directly inhibit apoptosis by blocking the activity of pro-apoptotic proteins, the executioners of cell death. Other viruses promote the expression of anti-apoptotic proteins, which protect cells from undergoing apoptosis. For example, the human papillomavirus, or HPV, which can cause cervical cancer, produces proteins that interfere with apoptosis. These proteins inactivate p53, a tumor suppressor protein that plays a critical role in initiating apoptosis in response to DNA damage. The study of viruses has been revolutionized by the advent of molecular techniques that allow us to analyze viral genomes, study viral proteins, and understand viral host interactions in unprecedented detail. These techniques have been instrumental in identifying new viruses, deciphering their replication mechanisms, and developing diagnostic tools and antiviral therapies. One of the most widely used techniques in virology is polymerase chain reaction, or PCR. PCR is a powerful tool that allows us to amplify specific DNA sequences from a complex mixture, even if they are present in minute amounts. Another essential technique is DNA sequencing, which allows us to determine the order of nucleotides in a DNA molecule. DNA sequencing has been crucial for determining the genetic makeup of viruses, understanding viral evolution, and identifying mutations that confer drug resistance. CRISPR/Cas9, a revolutionary gene editing tool, has also found applications in virology. Polymerase chain reaction, or PCR, is a fundamental technique in molecular biology that has revolutionized virology. PCR allows scientists to amplify specific DNA sequences, even in minuscule amounts. It's crucial for viral detection and diagnosis, especially when the viral load is low. PCR also helps monitor viral load, essential for antiviral therapy effectiveness. Coupled with DNA sequencing, it aids in viral genotyping, understanding evolution, and tracking new strains. PCR detects mutations for antiviral drug resistance. Viruses have been a constant threat to human health, causing diseases from the common cold to deadly pandemics. While we've made strides in understanding and combating viruses, the battle is far from over. New viral diseases and drug-resistant viruses highlight the need for continued research. Future efforts must focus on broad-spectrum antiviral drugs. Most current antivirals target specific viruses. Broad-spectrum antivirals would be a major breakthrough. Vaccines are one of the most effective ways to prevent viral infections. By unraveling viral mysteries and innovating, we can minimize viral threats and protect human health.